Hello everyone, and Marty, you are right, it takes a village and it was so much fun during the duration of this course. So hi everyone, we are the Above Below Seeker group. Our group acronym is SABRE, as you can see here, and it stands for Synchrony of Above and Below Ground Responses across ecosystems. And over the past 9 months, we have been exploring how above ground plant communities and below ground microbial communities respond together to global environmental change. And we are really excited to talk about our work today. Thank you for joining us. Next, please. So first, I would like to introduce our team. Abigail, Ashley, unfortunately, she couldn't be with us today because of a family emergency. We have Francis, McKinley, and I, Smriti. So we are a group of early career researchers. We are grad students and postdocs, and Ashley is a new faculty now. So last October, we met as a group at NCS, and our seeker mentors helped us pull our ideas together and diverse into new direction. So here's a picture of Carrie. She's explaining the diamond model of participation, which is very popular among the seeker fellows. And if you don't know about the diamond model of participation, it talks about how different ideas come together, generate discussion, and diverse in new and exciting directions. So our above and below team came together because of shared business interest and vision. Next, please. So before we dive into the presentation, we want to hear from you. Why do you think we should study above ground plant communities and below ground microbial communities together? So please react on Zoom. Give us some love if you think it's the first option. Give us some thumbs up if it's the second option. I can already see some smileys and confettis there. And you can also leave your ideas in the chat. There are lots of smileys. I can see love as well. Great. Yeah, thank you so much for your wonderful responses. And personally, I also think that we should study this together because they are in a relationship, but it is complicated. Next, please. So while studying these communities together, we can get great insights for the ecosystem. These communities are fundamental to fluxes of energy, nutrients, and matter of the ecosystem. So plants fix carbon and microbes recycle these nutrients. So they both are fundamental to the ecosystem. They also determine key ecosystem services such as food production and nutrient cycling, and both are important. And they are also temporally dynamic and interdependent. So the consequences of their association cannot be fully studied, fully known if we study them in isolation. So next, Francis will talk about how we access the connectivity of above and below ground communities. So Francis, take it away.
Hello, everyone. Well, we want to approach these association between above and below ground from two main concepts, which are coupling and synchrony. Coupling understood as the absolute value of the pairwise association among components of the ecosystem, in this case, above and below ground. We know that actually from literature, there is actually a kind of a mismatch between above and below ground, and actually that above ground, for instance, richness or diversity is not a reflection of below ground. But there is a huge variation globally in terms of geographically and how strong this association is. So that is something that we are interested in approaching. And we want to understand how these two components are coupled. And the other concept is synchrony, that is basically about the similarity in temporal fluctuations of these two ecosystem components related to a specific ecosystem function. And because of that, it has inherently a temporal component, so it needs to be sampled or researched a long time. Next, please. So when we think about how these might be respond to global change, there are some examples of other studies in these cases, particularly in response to nitrogen addition, and how different components of the ecosystem have certain degrees of coupling, but with nitrogen addition, this coupling starts to weaken or even disrupt to the point that the association is lost, and this would have effects on ecosystem functioning and services as well. Next. Also, we were expecting that, for instance, if we assume that under controlled conditions, above and below ground, both components fluctuate in synchrony, there is no data to know that, but we can assume that in controlled conditions, they can fluctuate synchronously. Then we would expect that in response to global change, that fluctuation, one of them or both of them can start to fluctuate out of phase. So there are changes in the amplitude, the time, or the frequency of the responses, and then they start to fluctuate asynchronously. And we would like to understand how these changes in these fluctuations and in the synchrony of those two components would affect ecosystem functioning, and how that would be, how they respond in terms of environmental variation in general, and how they would respond to particular climatic extremes. So now McKinley is going to talk about a little bit more about that.
Yeah, thanks, Francis. So we started out our data synthesis efforts with this kind of overarching question of how does the synchrony and coupling of these above ground plant and below ground microbial communities vary over time in response to a variety of global change drivers. And so as Francis sort of already hinted at, there's particular types of data that we need to be able to answer this question. We chose specifically to focus on plant and microbial community data, microbes being primarily fungal and bacteria. And so first off, they need to be spatially paired. We need to be sampling them from the same locations to be able to assess this coupling or connectivity. They need to be sampled at the same time. And then perhaps most challengingly, they need to be sampled repeatedly. And so some previous studies have kind of set a 4-year or a 4 growing season kind of repeated sampling as kind of the bottom threshold for an appropriate repeated timescale. But we know, of course, from the previous presentation that much longer timescales are much more desirable to be able to make sure that we're assessing these temporal fluctuations. And we also know, of course, that, for example, plant and microbial communities fundamentally operate on different timescales, and they can also respond at different paces to environmental change. So we opened up our interests to both observational and experimental field studies. And because we're interested in communities fundamentally, we were looking for any metrics that related to richness, diversity, or the composition or abundance of species that were present in sampled communities. As we mentioned, we're really interested in responses to different global change drivers. So we were also looking for climate variability data, primarily temperature and precipitation, but also really looking for data of extreme events, such as droughts and heat waves that we could potentially attribute some community responses to. And then, as Francis described, a lot of the connectivity of these communities is also very relevant for ecosystem functioning and services. And so we're interested in the functional traits or the different characteristics of these communities as well for plants and microbes. Of course, this information is harder to come by, especially for the below ground communities. So we're still figuring out where that can fit in, but we hope it will in the future. So we wanted to summarize a little bit of what our data search was able to compile so far. So first we tackled data searching in EDI and Google Datasets, primarily just using the standard search terms that one would imagine for looking for above below ground plant microbe community data. And we were able to comb through 180 data sets very closely. Out of that, only about 15% met those criteria that I described. It turns out that it's pretty hard to find not only the spatially paired above and below ground data, but also have repeated samples over time. And as has been the case for a long time, those below ground communities are generally much less studied than the above ground plant communities. Luckily, Neon was a really great resource for us. So we were able to collect data sets from all 47 of the terrestrial neon sites. And they have a really consistent sampling scheme for both the plants and the microbes communities. And so in the map on the left, all of those purple points are from the neon sites that we were able to collect from. And then so far, we have a small number of LTER sites that we were able to get appropriate data from. But we've been in contact with, I think, pretty much every information manager from an LTR site that we think would have data relevant to us. And we're going to keep having those conversations to see if we can find more data sets that meet our criteria. But you can see the LTR sites in the orange, and we had some co-located LTR and neon sites as well that are in the light blue in the map. And so, like I said, this is just a subset of really the LTR and the neon data sets. Those other 15% that we're working on combing through will add to that map and also expand into more of a global scale as well. But as of right now, we have a pretty good coverage of a variety of different ecosystem types. Right now, we have a lot of kind of forest grassland sites. Because that just happens to be where a lot of the neon sites are. But we are also really interested in the coupling and synchrony of these communities across a variety of different ecosystems. So we're planning on continuing to expand that coverage as well. Okay, so we wanted to give a very brief snapshot of the sort of analyses that we can do with this type of data. And so we have also targeted the Konza Prairie site. It has both LTR site and neon site present. And so we were using some of this neon data for the fungal communities and for the plant communities above ground. And so with these variance ratios, this is a way that we can measure the synchrony of these communities. Not saying the synchrony of the fungi and the plants fluctuating together, but their individual community fluctuations over time. And so when this is calculated, a value greater than 1 is suggesting that the community is fluctuating synchronously over time, and less than 1 would suggest that they're fluctuating asynchronously. So we see here that both the fungal and the plant communities are synchronous, but we can see that the fungal communities are over twice as synchronous, I guess you could say, than the plant communities. And so we have several sort of Konza experts on our team that we're hoping we can dive into this more with their local knowledge to sort of figure out why the fungal communities might be responding this way. But as I mentioned, we plan to expand this to many other sites as well, many different ecosystems to see how these responses change. Okay, and with that, I'm going to pass it off to Abigail to describe some of our other products that we've been working on.
Thanks. Okay, so one of our upcoming tangible products is an opinion paper. While we were going through this data collection search, we found that there was a big discrepancy between the interest and need identified by our mentors and other experienced researchers in the field, and the data that we were able to find that was publicly available to us, and that was being continuously monitored or collected. So we're writing an opinion paper as a call for coordinated monitoring of above and below ground community responses to global change, identifying this persistent data gap within the field, as well as formulating recommendations on how to move forward in building this robust data set. So as I said, this paper is addressing the gap in need for paired above ground, below ground community data for synthesis science. And we are also giving recommendations how to improve data collection as well as data availability for different practitioners such as conservation management, agriculture, and long-term researchers. And so we are a primarily US-based team right now. So we invited 4 different co-authors to collaborate with us on this paper. They have been in this field thinking about how above ground, below ground communities work together for a long time, as well as give a global context for what data sets might be out there, what opportunities might be out there for collecting this data. And we've sent a one-pager to Trends in Ecology and Evolution to see if they would like this paper published in their journal. But this is we're currently working on this manuscript and are pretty excited about it. And in addition to the pen and paper, we have several other products which focus on expanding our scientific community, expanding our scope beyond our small team as it is now. We would like to continue this collaboration and this effort beyond the 1-year Seeker course. So we wrote a Spark proposal through the LTR to continue our data search as well as expand our network beyond what we're able to accomplish as a team of 5. So you can see all the members that we invited to be part of the our Spark proposal team listed here. We're really excited to be collaborating with these people, whether it's through this Spark proposal or through other means, to continue gathering data and building a database of paired above below ground data sets that can be then used for synthesis studies. And one other product we're pretty excited about is we're hosting an ESA special session this year at the ESA meeting. This is a panel with, you can see it, the invited panelists here. We will be having a panel discussion about the panelist experience in working with above below ground paired community data, and then moving into a facilitated small group discussion for gathering ideas and recommendations of improved sampling methodologies, as well as continuing momentum forward, improving on the gap in spatiotemporally paired community data. So I'd love to invite all of you, if you're at ESA this year, we would love to see as many of you as possible participating in this panel. I think it could be a really cool networking event for early career researchers, as well as gathering context and experience from later career researchers. Okay, so with that, I'm going to move into some longer term goals following this really cool year of our Seeker course. Our first goal is to continue on building a more formal network, which will foster mentor-mentee relationships within the scientific community, all of who are united around this interest and need or belief in the need for a robust database of paired above below ground community data to get a full image of what's happening when communities are disrupted due to global change. And this will carry an emphasis on early career researchers and giving opportunities for people like us to start building our research in this within this field. We'd also like to establish a more formal above below ground database, which is a house for the data sets that we find within these next few years of our data set search, as well as an associated data papers about this search and the database itself. And lastly, looking to publish synthesis papers using the built database that will inform future experimental work and management. All right, so to finish up, I'll take a little bit step back, some key takeaways that we have from this really wonderful course that we've had the opportunity to participate in. The first is that data synthesis can lead in many directions. I think maybe each of our group members thought this final presentation would look different from each other and also from how it looks in reality. So I think this was a really fun just like process we've all taken together and taught us that learning and doing science is better as a group. And there's a lot more creativity that comes from working within a group. So kind of with that, how to be productive as a group doing science. Carrie was great, as well as all the other course instructors teaching us how best practices to do this. And here I'll speak for myself. I've really enjoyed collaborating with my team as well as all the others who are part of this Seeker course. And maybe one of the biggest takeaways our group specifically found is that reaching out and just asking for connections really builds better science and stronger communities. We've had super generous mentors, both, of course, those who were assigned to us through the Secret course, but also the people who responded to our cold emails saying like, we're interested in this. This is what we're doing. We read a paper by you that we loved. Would love to talk more. We've had many meetings that started out as that. So I want to say huge thank you to everyone who lent your time and expertise and aided us in this effort. And so kind of tied all together, science is better as a team. And that has definitely been the biggest takeaway from our experience with the Seeker course. So thank you so much to the Seeker core team who worked super hard to put this whole course together, specifically our mentors, Serita and Meghan. Thank you so much for your expertise and your time, as well as support from the LTR and NC's offices. So just a quick reminder, please join us at ESA if you're able to travel and able to come. We'd love to see you there, as well as take a snapshot of the QR code to visit our website. We'll be posting when our opinion paper is finished and published, as well as future proposals and future collaboration. And if you have any data you'd like to share, we're definitely still looking for that. So thank you very much for your attention, and we'd love to take any questions.
Awesome. Thank you all. We had a couple of questions in the chat, but maybe we want to just voice those out loud as well. Zoom has a new function that I'm so excited about. We can be in a webinar and actually allow the attendees to talk. So Meghan.
Can you all hear me?
Yep.
Great. It says it's talking. Okay. I thought the preliminary results, first off, this is fantastic. You guys did so much work and I'm just so impressed. So the Konza work, you found that the fungal species were much more synchronous than the plant communities. Now, my limited work with microbial data is like there are hundreds of species in these data sets. And I was wondering, are the synchrony measures sensitive to species richness? And if not, that's great. And then also just what was the size difference between the species pool of these two types of organisms? Thank you.
I'm thinking maybe somebody is talking but muted.
Hi, Meghan. Thank you for the question. And definitely the species pool was different. I don't remember exactly what magnitude. And I also don't know how that would like affect our variance ratio. So we'll have to check into that and we'll definitely update you on that.
Thanks, Smriti. Byron, you had a question also.
Yeah, I wondered about some of the biggest challenges in identifying these data sets, and I wondered if there were any sites that were like really stood out as models of good data management and good data availability.
I think I might have misunderstood your question, Brian, in the chat. So I think we had a lot of problems with EDI database. I personally had a lot of problems with database because of the naming convention as well, because some of the above plant communities database was housed with a different name than the microbial communities databases. But not to single out EDI, I think our question is novel in that sense. So people were not really thinking about paired above and below ground communities from the start. So I think that was really challenging for us. And McKinley and Abigail and Francis, if you want to add to that.
Yeah, I'll add that I think our biggest success was when we started reaching out individually to data managers. All the data managers we reached out to were super helpful, even if they had to say like, sorry, we don't have any data for you. But several data managers responded with like specific EDI links, which we weren't able to capture within our search, which maybe is kind of gets back to what Smriti was mentioning. But they also, more importantly, gave some like key insights of like, hey, if you wait a couple years, you'll hit your like 4-year threshold. Or I have a grad student working on this right now, but it will be like made publicly available within a few months. Stuff like that, that's just a lot harder to put on a database. So having like a contact per site that whose job it is to do this was really awesome and definitely where we were able to get the most success with finding data.
Yeah, I would second
Yeah.
that. I think we realized that a lot of above and below ground communities are sampled separately or they're put as data sets separately, but it turns out they were maybe sampled in the same bigger study or something that still made them suitable for our purposes. It was just a matter of finding them. And so that's one thing that we're hoping that we can pursue more with the Spark team, fingers crossed, is that we'll have a lot more folks able to work on that and like find those connections because it exists. We just have to find where it is and then encourage more in the future.
That's really interesting.
Yeah. And in my side, I was doing the search on Google dataset search, which goes like, is the scope beyond all the other repositories? Is more global. The challenges was like finally, like we were focusing mostly on field studies, not in greenhouse studies, because we want at least the plant community to be like as intact as possible, or at least like an actual plant community. And the challenges, one is most of the data is only for one sampling point for below ground, even though above ground they can have like several sampling points. So that mismatch in the sampling scheme between above and below ground was a challenge. Another one, below ground has data in different formats. So some of them provide OTUs or the genomics stuff, and something that goes beyond my purview, so people help me more. But those things made it a little bit difficult too. Other things are people sometimes in different parts of the globe, they do have, like we have to go through all the methods in each of the papers for the data sets. And actually they do have unpublished below ground data, but for some reason they don't publish above ground data. We try to reach out to the authors because they state in their papers like data available upon request, and most of them they really don't reply, which is kind of like why. So that is another way of making really hard, is like people are in other parts of the globe, they're not used to actually putting their data on repositories, or some of the data, but not all of it. Sometimes it's just a summary of a table of the data, but not the raw data itself. And when you try to reach out like, hey, we're doing in this synthesis, we're interested in compiling this data, we were interested in maybe finding if you have this and this data available, and people just don't respond. So that part of upon request is really hard to reach out. So yeah, it's the different ways of people do things in different places that makes it really hard to reach out and make it available for everybody. And that is one of the goals of the group, is actually trying to compile as much data as we can and make it available for the public.